Welcome back, everybody. We've got, not quite, I guess we've got a quorum here, so we'll get going. So what we're going to talk about here is we're going to go through three different components in this finishing up. First, I want to talk about the gut-brain connection, the enteric nervous system. Within GI2019, we've talked about digestion and intestinal permeability. We've talked about the gut microbiome, and we've talked about inflammation, but we haven't really talked very much about the gut-brain axis. It's been alluded to on several occasions, but I want to dig in a little more deeply and talk about this incredible relationship and what it means in some of the studies that have been looked at. And from there, I want to take you into a deepening of our own gut feelings and connect into that, and then move into a case study. Just again to explicate, how we use these tools, and it's all of the tools. It's our go-to-it operating system. It's the timeline. It's the matrix. It's the understanding of antecedents, triggers, and mediators, telling the story, the importance of that, and then looking at what we can expect with our patients who've been on a long and winding road of moving from ease to dis-ease, and that it's not necessarily going to get better all in 4 weeks on a new diet, but rather it's a winding road back from dis-ease to ease in the process. And then we'll close with just a recapitulation of the highlights of where we've been in the process. So, it's generally understood and accepted that a stable gut microbiome is essential for physiology, and it contributes to the appropriate signaling of the gut. So we see that our behaviors, our emotions, the inflammation that's going on, what's happening with our gut microbiome, that is going to be signaling to the brain. And that when there are imbalances that are going on, that is going to give a different set of signals to the brain when the microbiota is. So I'm going to show you 3 slides that kind of progressively go into talking about the neurotransmitters like serotonin that's giving input to the brain, the cytokines that are released from imbalances in the microbiome that are giving signals to the brain, and the metabolites from the bacteria that are giving signals to the brain. All of this input is going forth and is gathered in the brain. And so here we see an increasingly in-depth graphic which shows on our left the serotonin, again, the neurotransmitters, the cytokines, the bacterial metabolites. But in addition to that, we also see that there is an afferent nerve that is feeding information up into the brain. And where it goes in the brain is into the area first of the limbic system, of the amygdala. Recall that the amygdala is all about processing emotions, memories, and motivation. It's a reaction to stimuli that are happening. So let's take this just for a moment and consider, the input that's going from my gut is going up into my brain and is being processed in the amygdala, through my system that is processing emotions and memories. Now, the other area that we'll see right here is not only is it going to the amygdala, but it's also going to the insular cortex. So again, we see that there are, cytokines, we see that there are gut peptides and metabolites, there are neurotransmitters, there's vagus nerve input that's all going into the brain. And where it goes into the brain is into the area called both the amygdala and the insular cortex. The insular cortex, which is a small area in the cortex, is providing the emotional context for our physiologic experiences. That is, this is the part of our brain that gives meaning to what our bodily experiences are. Think about that. This gives meaning to what's happening in our body, and it's gathering the information from our gut. That's where the information goes. So we start to think about, well, what happens as we perceive pain? How does it integrate our basic emotions? Addiction and desire. The sense of body, interoceptive awareness that's going on. All of these components, our sense of self, reading social cues. Another way of saying that is, these are our gut feelings. Think about it. The input that we're getting from our environment all around us is integrated through the gut and being sent to the brain. And what's driving that awareness? What's driving those metabolites, those inflammatory cytokines, those neurotransmitters, the vagus nerve activation? What's driving that? There's a single word. Should we all say it in unison? The microbiome. So I like to think about the macrocosm and the microcosm and what's happening in the world and all the things that are around us, and we see alterations in the microbiome of our world. Some people have asked me about some other training that I have with indigenous cultures in Mexico, in the Sierra Madres, where I've been going and studying for 18 years. And we find that the indigenous people, 5% of the people in the world, who have stewardship over 20% of the land, that land holds 80% of the biodiversity of the world. And we continue to be losing our biodiversity in the world and in ourselves. And I'll talk about those interrelationships that happen, but I want to bring it back to ourselves, and that is the gut feelings that are going on. So what we see is that information is gathered from the gut, from a relationship of, and they now call it, I don't like the acronym of GBM because it makes me think of glioblastoma multiforme, but the gut-brain microbiome axis of the interrelationships that are going on. So that information of what's happening with our microbiome is integrated in our brain through our emotions. It's connecting to our memories, and then it's going through and signaling both through the vagus nerve going back down in terms of our balance between parasympathetic and sympathetic drive, and also through our hypothalamus and pituitary glands, which is the major controller of all of our hormones. All of our hormones are coming through this. So now all of a sudden we begin to get a sense of, I wonder if the foods that I'm eating and their effect on the microbiome is affecting everything that's going on in terms of my mood, my feelings, my reactivity, my anger, my cravings, my addiction, my sense of pain, my sense of self-worth. It's like, whoa. That's actually happening and being mediated through our gut microbiome. Now, I've been studying this aspect of the microbiome and its interrelationship with health and well-being for now almost 30 years. I would say 28 years that I've specifically focused on it. But as I dove into this, I was asked to give a talk about the gut-brain axis, and I dived into this literature around how these pieces fit together. And I was literally astounded at how much research we have and how profound and where the connections are. I don't know if this works for any of you, but looking at just this slide and what I'm talking about in terms of gut feelings, it activates me in a way that is far beyond what I would have imagined in terms of the role of our microbiome and the role of our gut. And recall that while I'm focusing on the gut microbiome, that's going to be informed by how well is digestion working, how permeable is our intestinal lining, what's happening with our immune system. So the interrelationship of DI-GIN is happening all the time. A lot of it may be mediated through the microbiome, but all of these functions that are happening in the gut are essential. So we can also see that not only, as I've said several times, I think, there's 10 times more information going from the gut to the brain as there is from the brain to the gut. But that information that's going from the brain to the gut, in terms of the vagus nerve and a sympathetic overdrive and the role and relationship of stress, that causes our beneficial bacteria of Lactobacillus and Bifidobacter to be depleted, to be suppressed. That causes our secretory IgA that we've talked about, that is an overall indication of our immune health and well-being, to be suppressed. The catecholamines of epinephrine and norepinephrine are going to cause these gram-negative bacteria that contain lipopolysaccharides that are going to stimulate NF-kappa B formation and inflammatory drive through the toll-like receptors that you've heard myself and Jerry talk about, leading to increases in Yersinia species and Pseudomonas species that are there. Studies from the Russians have told us that anger and fear actually increases the specific species of Bacteroides fragilis, one that in combination with other gram-negative bacteria that produce lipopolysaccharides can cause an inflammatory reaction. Now, we continue to be looking at there is a low-grade inflammatory response that's going on by alterations in the gut microbiome that we're not yet able to measure effectively in the serum. And that doesn't come out in our fecal inflammatory markers like calprotectin or lactoferrin or eosinophilic protein X. But we can see it by alterations in the gut microbiome. And we're moving in the direction of being able to define the patterns that are associated with a pro-inflammatory microbiome. And yet, you don't actually need that information because you know what information is driving that pro-inflammatory microbiome. And it's, of course, the foods that we're eating. Now, we've talked about therapeutic diets of using a Renew diet or an elimination diet or an elemental diet or a low FODMAPs diet therapeutically to help our patients move along. But then there are also the maintenance diets. And we have some of these tools in the toolbox, but looking at a Mediterranean diet, looking at a whole foods diet of the foods as they're coming from the earth, while listening to what those food sensitivities may be for a given individual. That's really where we want to be going. And yet, we find that, things like sugar and alcohol and other things will tip us over so quickly and induce changes in cravings. And they're being mediated through the gut microbiome. And so we can see that the cravings as well as the moods and the behavioral issues are directly related to the microbiome through the metabolites that are produced, through lipopolysaccharides and cytokine production, through neurotransmitters like serotonin, through the composition of macronutrients and micronutrients, and through the bacterial metabolites. These things have been shown, as in this study from Bursick, where you can actually then use agents like Bifidobacter longum to be able to induce a GABA-like response that suppresses what's going on and decrease depression. So here we see a number of different microbial species like Saccharomyces that causes changes in norepinephrine. Streptococcus and Candida will increase serotonin production. Serratia will decrease dopamine production. And lactobacillus will have an effect on acetylcholine production. So as we go through this, we begin to use the term, as has been described, of psychobiotics. Psychobiotics are those organisms that are probiotics, but that they produce a health benefit in patients who are having some kind of neurologic, mood disorder, or psychiatric-related illness. Now, a lot of the research that's been done so far has been done on rodents, on rats and mice, and looking at seeing an antidepressive and an anti-anxiolytic effect that's going on. There are some human studies that have been done as well that help us to understand. So here's a human study that was a randomized controlled trial looking at 40 individuals, and they used this combination probiotic with several different strains of Bifidobacter or several different species of Bifidobacter with substrains and several different species of Lactobacillus. And what they found was that when there was a Leiden scale of a depression severity scoring, that it was significantly reduced versus placebo in those who received this combination probiotic. Another study, 47 people, again, looking at a specific individual, Lactobacillus casei subspecies shirata. And what they did was they were looking at medical students, a good guinea pig to be able to use in this situation, and they were seeing how well did they do in response to the psychological issues of taking exams. And what they found was that there were significantly higher amounts of serotonin in the population that was receiving this particular probiotic, this Lactobacillus probiotic. Another Lactobacillus single strain probiotic was looking at athletes and saying, when we look at them and we give this specific piece, we found that their moods were improved and their NK cell function, their natural killer cell function activity was significantly increased, and their fatigue was decreased when they took this probiotic. That was, again, a placebo-controlled trial. So here we see a number of different trials in humans that are saying when we're giving probiotics, in two cases, specific lactobacillus strains. In another case, a combination therapy of Bifidobacter and lactobacillus. What we found is it's able to make a difference in the overall mood and psychiatric functioning of these individuals. So this is really quite profound. And as we come back and we want to look at, well, let's step back again and look at, well, what are the functions of the gut microbiome? So there's metabolic activities, and we've talked about these. We've talked about the short-chain fatty acids and how the microbiota is used to actually be able to help synthesize specific vitamins that are necessary. It's used to be able to produce energy, such as N-butyrate. 70% of the ATP that's produced in our colonocytes comes from the butyrate that we get from the fermentation of bacteria and their interaction with soluble fibers that's going on. We look at the trophic activities, that is, what is the activity that helps it to heal the lining of the gut? The microbiota is necessary for helping not only with immunomodulation, but also to be able to heal that intestinal permeability that's going on and allow for epithelial cell differentiation to occur. Immunologically, we see that there are a series of effects in terms of decreasing cytokine production, decreasing inflammatory cytokine production, increasing anti-inflammatory cytokine production, increasing secretory IgA, creating a barrier function, and competing with potentially pathogenic organisms. So there's a number of different roles and ways. It's also, I want to come back and remind you that the immune system is part of the microbiota is part of the education of the immune system in the first place, over the first 2 years of life. I mentioned that early on. And there are all these protective effects that are working to, compete for nutrients, to compete for space, to compete for adherence. Creating the biofilm layer that is just right, not too thick to cause problems, not too thin to increase permeability issues. And then there's also this bidirectional interface that I've talked about between the gut and the limbic system and the insular cortex and our sense of self and all of our feelings, as well as stimulating back through our vagal nerve and through our hypothalamic pituitary axis. So to me, having studied this for a long time, it's amazing to me the depth of what is happening here. And what we find is that, when our system's in balance, when we have the right amount of symbionts and pathobionts, as good guys and bad guys, we do well. But so much of the time we have a relative imbalance, and that relative imbalance is either because an insufficiency of the good guys or an excess of the bad guys that are there. But we're not trying to just focus on that. We're trying to focus on how do we bring the system back into balance. So you've heard me talk about how the gut flora works to inhibit pathogenic bacteria, how it works to improve epithelial function overall. And as it's improving epithelial function, it's doing that through these short-chain fatty acids, but it's doing it through producing mucus. It's doing it through increasing the barrier function. We haven't talked about, heat shock protein 25 and its role in being able to be protective and helping the epithelium to heal itself. There's all of the cytokines over on the immunoregulation side that we look at. So there's all these different functions that our gut flora is playing. And there's a crosstalk that's happening all of the time between our gut microbiome, our immune system, and our central nervous system. And so these interrelationships, that when our immune system is altered, that it's sending cytokine signals to the brain. Dr. Mays and others have talked about the inflammatory aspect of depression and of anxiety, and that when we see these inflammatory cytokines, they're actually having an effect on overall mood that's going on. Some of that is going to be mediated through the microbiome, but some of it's going to be mediated through both the metabolites and the inflammatory cytokines that are being produced. And there are microbe-derived molecules, not only the short-chain fatty acids, but the tryptophan metabolites and other neuroactive molecules. A piece that I'm working to investigate right now and learn and understand more completely is how do we begin to evaluate the microbiome metabolites and really see what effect are they having overall in our body. Because there are more metabolites from the microbiome than there are metabolites from the human genome. That would make sense when we talk about the difference between 1 million genes that we have in our gut from the bacteria versus 23,000 genes that we have in each of our cells. So there's a difference that's going on. So we look at these pieces, and in the immune module they talk a little bit more about the molecular associated or the microbe-associated molecular patterns, the MAMPs, and the pathogen-associated molecular patterns, the PAMPs, and what impact they have on stimulation of the toll-like receptors and on Th17 differentiation and production of nuclear factor kappa B. There are many different elements that are going on here. What the focus is to understand, let's look at the crosstalk that's happening. And so as we begin to understand now the N of dig in, the enteric nervous system, it's controlling our motility. It's controlling the blood flow. It's controlling what nutrients are we actually taking in. It's controlling what is happening with the inflammatory process. So all of these aspects of, if you will, the gut-wrenching experience, or having butterflies in your stomach, or relying on gut instinct. We just look at our language, and in our language we know that what's happening in the gut is informing what is happening in our central nervous system and our relationship to the world at large. So we know that the feelings that we have will also trigger reactions in our gut. Now, this is talked about. There's a couple of books that I found to be really useful. Emmerin Mayer. Emmerin is an amazing MD, PhD who's at UCLA, who's been talking about these ideas and really taking a systems biology approach to irritable bowel syndrome for quite some time. And then this book by John Crinan and Ted Dinan, who are from Cork, who are from Ireland, talking about these ideas, who have brought this idea of psychobiotics into the mainstream of our GI colleagues and being able to talk about it. Because of the input that's going from the gut to the brain, and then we also have the input from the brain to the gut that is going to affect motility and visceral perception. Now, when we have inflammation that's going on, even a low-grade inflammation, and we have those release of, say, calprotectin or some other molecule that's going on, it increases the activation of the afferent signal from the gut to the brain. So visceral pain perception is going to be influenced by co-emergent inflammatory processes that are going on. And so again, we see this two-way interaction that is going on. Our stressors, as we've talked about, such as stressors from over-exercise, will lead the vagus nerve to cause changes in our intestinal permeability. It's happening in both directions. So in this gut-brain axis, we're seeing that the gut is using its microbes, its metabolites, its cytokines, its neurotransmitters, its afferent vagal nerve stimulation. All those things are going to the brain, and we're receiving neurotransmitters, we're receiving vagus nerve input, and we're receiving hormonal input, all of which are having an effect on the gut, coming from the brain. Again, looking at this particular slide is one that is very, very important to me. And when I'm talking to patients about what's going on with their moods, I'm wanting to help them understand, like, this isn't just all in your head. I'm not telling you that you just have to think differently. So when I have the 43-year-old patient with severe anorexia, presenting to me, at 50 pounds from severe issues with food, everyone wants to give her the diagnosis of anorexia, but with chronic idiopathic constipation and pain that she's having all the time, anytime she eats anything at all, it's a struggle for her to be able to add the foods and gain back. How do we work with her microbiome to get it back into balance? How do we work with the chronic constipation that's going on to help it so that she doesn't have pain? And how do I use tools like what is called dynamic neural retraining, DNRS system, or biofeedback tools that will allow her to have the neuroplasticity to start developing other pathways, to not have the same way of thinking and relating. Well, I come back to the insular cortex and say, whoa, there's a place where we can do it right there, and to the amygdala. Let's look at your memories and your emotions, and let's begin to work with a process that is saying, we're going to start to set new neural pathways. So when you have that input that's coming in, you're not interpreting it in the same way as you have before. So these are all kinds of tools that we have in our toolkit to be able to help. So we're helping with the food. We're helping with dealing with the motility issues. We're helping to deal with the awareness issues of body sensation. And we're using tools that are not only dietary and nutritional supplements, but we're also working with the way in which we think about things, the way in which we relate to things, the way in which we perceive things. All of these factors are going to make a difference, because ultimately I want to be able to help modulate this gut-brain axis. And there's also some probiotics that now there are studies that are being demonstrated that are actually helpful. So we talked about the study with Lactobacillus casei shirota, that actually was able to demonstrate a reduction in stress in a placebo-controlled human trial. Or the combination of lactobacillus and bifidobacter from the earlier study that I showed, that was able to demonstrate improved cognition. So we see that there are a number of different studies, and here we see from the Journal of Microbiology last year talking about just some of the studies that have been done and ways to do this. Now I want to highlight again, when we're focusing on this, we're focusing on specific probiotics for specific therapeutic interventions. This is not going to one size fits all. We don't say, here, I'm going to give you an antibiotic. I don't have any idea which antibiotic it is or what it does, but it's going to help you. Same way we would never say, here's a probiotic. I have no idea what it actually does, but it's going to help you. No, we have specific species and strains that we use for specific therapeutic interventions, just as we have specific antibiotics that we use for specific interventions. And we also know and recognize, one of the things we've tried to underscore in the conversation is that the probiotics that we give are transient. They're placeholders. They help make a therapeutic change for the duration that they're being given. And they can induce some alterations in the microbiome itself, but they are not permanent. We want to use them for specific clinical indications. And as we've said many times, it's diet that makes the biggest difference. The diet, which is giving the prebiotics to allow the right bacteria to grow, is the essential aspect. I mentioned yesterday, just briefly in passing, but there is now new literature that is showing that exercise, independent of diet, is also able to help modulate the immune system. And some newer studies looking at the stress response are saying that when we move into a deeper state of relaxation, of rebalancing what's going on, we can also modify the gut microbiome. Well, this would make sense from the data I just showed you of stressors decreasing lactobacillus and bifidobacter, of anger inducing changes in gram-negative Yersinia and others. Was it Pseudomonas? I can't remember which one it was. But our feelings and our awareness and our stressors are going to have an effect on what's going on. So while the things that are on this slide seem like, yeah, I know that. That makes sense. I got it. But how do we actually slow down and begin to relax? How do we take the time? I like to share an awareness I had one time. I like to give Michael Pollan's book Food Rules to patients. It's 64 food rules. It's a very short book. It's like this big, and there's basically 64 chapters. Each chapter is less than a page, and it will have a saying on it, and then just a little reading about it. And so I was sitting at my desk at my office one day, and I was eating my lunch and had the computer on in front of me and was looking at some things and picked up the book. And I with books like that, I like to just flip them open to a page and see, well, what is it telling me today? And I opened it up and it said, always eat at a table. ... A desk is not a table. And I looked at it, and I read it, and I looked at my food, and I looked at my computer monitor. And in my office, I have a little round table that I'll, have for like conferences and talks with people. And so I'm like, okay, shut off the computer, take my food, go over, sit down. It took a half an hour to eat my food. 3 people came in during that time. Is everything okay? What's wrong? It's like our culture is where are we at? But it actually slowing down, mindful eating. Another story I like to tell is I like to teach patients the raisin meditation. How many people have ever done the raisin meditation? Where you take a single raisin and basically you really connect to it. You smell it, you listen to it, you touch it, and it takes about, when I do it, between 20 and 30 minutes to eat that single raisin. Well, when you bite into that raisin after all that anticipation, just a single raisin, it just fills your mouth with taste. And you're like, wow, one single raisin. So I've done this. So here I was one day, we live on an old farm, and I was out on the tractor, and I was all hot and sweaty, and I came in, I was hot, I opened up the pantry, I'm like, I need something to eat. And it's like, oh, there's some raisins. And so I took a handful of raisins like this big, and I shoved them in my mouth all at once, because I was just going to walk back out and get on the tractor. And I bit down, and all of a sudden, oh my God, raisin meditation. I had a whole mouthful of raisins, but I had a taste in my mouth. That was like, it reminded me of the raisin meditation. So then I'm just like, okay, I'm going to go and sit down at the table and just take like 15 minutes to chew this whole mouthful of raisins. My point is, it is never too late to be mindful in eating. So next time you eat, consider that. And if it comes through your mind and you're looking at your plate and you can't even remember eating anything and there's one bite left, be mindful with that. We don't do that. We need to do that. We need to slow it down. Now there's other aspects, and I use tools in my practice, including heart rate variability biofeedback. I use a brainwave biofeedback tool. I'm trying to use whatever methods are possible that someone is open to. Some of my patients, we have deep conversations about prayer. Others, we talk about yoga, or we may talk about meditation, or we may talk about qigong, or if they're not into any of that, we may use some kind of app-based tool for biofeedback to help them. I don't care what thing it is that they're doing. It's like I've got to find and listen to what matters to them and what's going to touch them. And I'm asking about that each time I talk to them. So we find that, overall, we want to be taking all of these aspects into account. And I've highlighted that the effects of our lifestyle, the processed foods, the food additives, the emulsifiers, the lack of phytonutrients in what we're eating, the lack of isoflavones and polyphenols, the lack of essential omega-3 fats and omega-9 fats, the presence of trans fats, the antibiotics, the stressors, the tobacco, the other drugs, the sleep disruptions, the sound, TV, social media, all these aspects. Well, we have the ability, we have antidotes to be able to work with them. Not only by focusing on whole foods, but also by focusing on our relationship to the environment. It's fascinating when you read the Chinese literature or the Japanese literature on forest bathing. How many people have heard this term forest bathing? Pretty good, more than half. And so we see that just going out and being in the woods, being in nature, increases our NK cell function, increases our immune function, decreases our risk of developing disease. Finnish study was done looking at the microbiome in relationship to where we lived and found that those people who had an atopic illness, that the group of people who lived in the city and didn't go out, their microbiomes were different and their risk of having atopic illnesses was highest. People who lived in the city near a park had a different microbiome and their risk of developing atopic illness was lower. People who lived in the city and spent time in the country had a different microbiome, and people who lived in the country had a different microbiome, and the risk of atopic illness descended through each of those areas in a controlled study. This is done in Finland. Some of the most coolest research on microbiome is done in Finland, but it's about where do we spend our time? I'm going to be giving a talk in January. I don't have the slide in front of me here, but I want to highlight I've been using a tool to look at my own heart rate variability, and as I did that, I found like, wow, you're a lot more stressed than you present yourself to be. You know, like you wound pretty tight, traveling, doing all these things. So I'm like, okay, I'm going to double down on my diet, which I did, and went keto and lost some weight and did well. And then I said, well, I need to focus on sleep, and so I started really focusing on getting 8 hours of sleep. And that didn't change my heart rate variability. And then I said, okay, well, I've had a meditation prayer practice for a long, long time. I'm going to spend more time doing that. I'm going to do some brainwave biofeedback. And that didn't change my heart rate variability. And then, kind of not as part of my self-experiment, but part of what was happening in my life, is I needed to get outside in the wintertime. And I didn't have very much energy. It was the end of my chemotherapy, and I needed to get outside, and I started going outside and just playing outside like a little kid. And I would go play in the stream, and I would just move water around, and I wasn't hiking, I wasn't trying to get exercise, I wasn't trying to do anything. And then I started noticing, oh my gosh, my heart rate variability has like doubled over this period of time. And all of a sudden, just being outside and being connected fundamentally changed. Now, as chance would have it, I did a repeat of that metagenomic study that I had done, and I saw changes in my microbiome as well. So this is just an N of 1. It doesn't really mean anything, but it means something to me. Because what I found is that by slowing down, and at that point in time, my diet hadn't changed. It was still pretty consistent with what it has been, which is kind of a low-carb pescatarian diet. And yet, there were fundamental changes that happened for me, and changes in my inflammatory cytokines, and my transforming growth factor beta-1, and my HSCRP. All those changed. So I know that this is real, and I'm highlighting it because it does make a difference. And so, connecting to the world that we live in, connecting to the, if you will, the indigenous microbiota that's in nature makes a difference. The diversity around us and the diversity within us are connected, and they're connected to our immune system. So everything that we eat, everything that we drink, everything that we touch, are animals that we have. We like to make fun with Dan about his penchant for dogs, but the relationship with the animals that we have helps to change our microbiome as well. Who took the chair that I had up here? Well, we'll do it this way. We're going to just take, I want to just slow down for a minute. Oh, great. Thank you, Alex. Did you take my chair? No. Thanks. I'm going to just take a moment, a few moments, and be able to slow down before our last case. And what I'm asking you to do is to sit comfortably at home. Not too comfortable, not laying back, but just sit comfortably with your back, your vertebrae kind of straight. Think of them as coins stacked up. Close your eyes. And you can, if possible, you bring your tongue to the back of your teeth on the upper palate. I'm going to ask you to breathe in deeply through your nose and out slowly through your mouth. As you do this, I'm going to ask you to put your hands on your belly, on your tummy. And as you breathe in, I want you to feel your tummy pooch out. And breathing out slowly through your mouth, feel your tummy go back in. Just allow this breathing in, feeling your tummy expand, breathing out, contracting, allowing you to move into a place of relaxation. Breathing in, breathing out, just slowing things down. You'll notice that there's thoughts that come up. That's the nature of the mind. Don't try to grab your attention, but just allow yourself to bring your attention back to your breathing and your tummy, breathing out. What I ask you to do now is, as you're breathing in, feel a breathing in of not only the oxygen, but of light and all the energy around you, and blowing out. And with each breath in, feel that filling your torso, filling not only your chest, but your tummy with light. And bring your awareness to what's happening in your gut right at this point in time. Breathing in slowly through your nose, breathing out through your mouth. As you do this, I ask you to bring your attention to your gut. And listen. Listen with your hands. Listen with your ears. Listen with your mind. What is my gut telling me right now? Oh, I shouldn't have eaten those eggs. Oh, I feel filled with all that I'm receiving. What are you feeling right now? Knowing that other sounds are going to come into the room and the mind is going to drift, and why are we doing this? And, I've got to get home in an hour. All those thoughts will come up, but just allow yourself to focus on your breath and to be able to listen to what arises. Some people might call this intuition. Some people might call it gut instincts. But just listen to what is arising. In my mind, in my awareness, I see and feel a tunnel. A tunnel that's filled with many different characters from my life who are supporting me on my journey. Allow yourself to listen to whatever images arise, whatever words arise, and allow yourself to just take those in. Don't analyze them. Don't try to figure them out. Just be aware of their presence. And offer thanks and gratitude to your gut for all that it does in connecting you to the world, in connecting you to yourself, and offer that gratitude and thanks. And just pat your tummy with a thank you. And bringing your hands back down to your lap. Breathing in deeply through your nose and out through your mouth. We'll do that 3 times. And allow yourself slowly then to open your eyes and come back to this place. And recognizing with a rejuvenated body, mind, and spirit, we'll come into the last bit of our conversation and teaching during our course.